If you love horror movies about alien parasites taking over hosts, brace yourself. Our own planet harbors something just as terrifying. Meet Ophiocordyceps, the zombie ant fungus. It doesn't just infect ants, it runs a masterfully precise spore dispersal strategy. Once spores breach the exoskeleton, they flood the bloodstream and release bioactive compounds. Fungal cells multiply, weaving an internal network that, paired with those chemicals, acts like a secondary nervous system that overrides ants' muscle control. It forces the ant to climb exactly 25 centimeters up a plant, precisely where forest humidity and temperature are ideal. Then comes the death grip. The fungus compels the ant to bite down hard on a leaf vein, lock its jaws, and then kills it. Using thread-like mycelium, it anchors the corpse, then sprouts a downward stalk topped with a fruiting body. It contains spores that mature in pressurized sacs called asci. When internal pressure peaks, the sacs rupture, explosively launching spores into the air. Positioned directly above foraging trails, the carcass becomes an organic sniper tower. Spores rain into a targeted kill zone below. Healthy ants passing through will get coated. Fungal enzymes breach their exoskeletons, invade the bloodstream, and restart the cycle. Through evolution, one sacrificed host becomes the ultimate reproductive weapon. The fungus Mesospora cicadina doesn't just infect periodical cicadas. It manufactures addiction and hallucination. It produces a potent chemical cocktail, cathinone, an amphetamine-like stimulant, sometimes laced with psilocybin, the hallucinogen found in magic mushrooms, flooding the cicada's nervous system. These compounds induce hyperactivity and numb the insect to its own gruesome disintegration. As the fungus consumes the abdomen, it replaces it with a chalky, spore-packed plug. Drugged and oblivious, the cicada flies normally. Wing clicks and movement crumble the plug, raining infectious spores onto the forest below. But airborne dispersal isn't enough. The fungus hijacks mating behavior for high-efficiency transmission. Infected males retain their species-typical buzz to attract females, but also adopt the female wing flick signal. Healthy males, deceived by this hybrid display, rush to mate and collide directly with the infectious spore plug. A psychedelic strategy. The fungus turns its host into a tireless, disintegrating zombie that actively lures new victims into infection, ensuring its spread across the brief, synchronized emergence of its 13 or 17-year generational cycle. The horsehair worm hijacks crickets for a water-bound reproductive mission. Larvae enter indirectly. They are consumed by aquatic insects, which die on land and are later scavenged by crickets. Inside, the worm bores into the body cavity, growing coiled to 3 to 4 times the host's length while the cricket appears perfectly normal. When maturity demands water for mating, the worm deploys molecular mimicry, secreting proteins that mimic the cricket's own neural signals to sabotage its behavior. It reverses natural negative phototaxis, the instinct to seek darkness, and drives the cricket toward bright, open spaces, triggering false sensations of heat and dehydration. And then the final trigger arrives. Water's horizontally polarized light reflection, normally avoided, becomes irresistible. The manipulated cricket stumbles toward the shimmer and plunges in. Water contact cues the adult worm to uncoil and burst through the abdomen, emerging to mate. A chilling masterclass in parasitic behavioral control. The Costa Rican parasitoid wasp commits a chillingly precise feat of mind control. It hacks its host's instinctive blueprint. A mother wasp paralyzes an orb weaver spider, glues an egg to its abdomen, and departs. The hatched larva feeds on spider's blood for approximately 2 weeks while the spider behaves normally. When it's time to build its cocoon, the larva injects chemicals that sabotage the spider's 5-step web-building program, deleting the scaffolding and sticky spiral phases. The drugged spider becomes trapped in an obsessive loop, reinforcing only anchor lines until they're 20 times thicker than normal. The result, a stark, simplified fortress of heavy cables meeting at a reinforced central platform. With construction complete, the spider sits motionless at the center. The larva then kills and discards its host, spinning its own cocoon suspended in the weatherproof structure. A masterclass in targeted behavioral corruption. Not teaching new skills, but hacking existing neural code to construct a custom nursery for the next generation. The lancet liver fluke executes a brutal strategy. One parasite dies so thousands of its clones can thrive. An ant ingests multiple flukes via snail slime. Most flukes create protective cysts safely in the abdomen, but one, the brain fluke, migrates to the head, positioning itself near the subesophageal ganglion controlling the mouthparts. This lone fluke is a suicide operative. It cannot form a protective cyst and will die when ingested by an animal like sheep, but its sacrifice ensures its siblings reach the sheep's liver to reproduce. Manipulation is temperature-gated. In warm daylight, the fluke lies dormant and the ant behaves normally, avoiding lethal heat exposure. At dusk, cooling temperatures activate the fluke. It manipulates the ant's nerves, compelling it to climb vegetation and lock its jaws onto a blade of grass at the tip. Suspended like bait, the ant awaits grazing animals. If untouched overnight, in the morning when it begins to warm up, the fluke releases the ant. The ant descends and rejoins the colony, unaware that it will repeat the climb at twilight when temperatures drop. Eventually, the ant is consumed with grass by a grazing animal, delivering its siblings to infect the host liver. But that single brain fluke will perish, lacking the protective cyst needed to survive. If you thought the last few were bad, prepare yourself, because this one takes the crown for sheer bizarre theatricality. The jewel wasp performs something more sinister than mere parasitism: precision brain surgery to hijack a cockroach's will. She ambushes a cockroach, clamps its neck, and then delivers a thoracic sting, injecting venom-rich in GABA neurotransmitters to temporarily paralyze its front legs. This buys time for the critical second strike. Using her stinger equipped with mechanoreceptors to feel through tissue, she navigates into the head cavity to target two neural clusters controlling motor function and escape behavior. There she injects venom that blocks octopamine, the neurotransmitter governing the urge to flee. The roach isn't paralyzed. Its will to escape is erased. Now docile, it grooms passively while the wasp trims its antennae, both to drink hemolymph for energy and to create a stiffer, easier-to-grip leash. The wasp walks backward, leading the compliant roach to her burrow. She seals the entrance with debris. Remarkably, the roach could escape but doesn't. Its survival instinct chemically deleted. Inside, a single egg hatches. The larva consumes the roach alive over days, strategically sparing vital organs until last to keep its meal fresh. A chilling masterpiece of targeted neurochemical control.